there's still people steadily trickling in, so we might wait just another minute or so. While we wait for that though, so there's no general updates or announcements on behalf of the Biocurator working group team, but is there any general announcements from anyone else on the call? If not, I think we can get started. So our working group call today is continuing our series of sort of working through some of the different clinical domain working groups and expert panels within ClinGen and just sharing their experiences to date, challenges they've faced and where they're at. So today we have the Syndromic GCEP co-chairs, Dr. Alison Coffey and Dr. Anne O'Donnell-Luria presenting today on behalf of the Syndromic GCEP. So if there's not anything else that anyone quickly wants to add before we jump into it, I will hand over to Anne and Alison.
Thank you, Alicia. And are you going to share the slides? Perfect. Thank you so much. So thank you, Alicia, for the introduction. Yes, we are going to talk to you today about the ClinGen Syndromic Disorders GCEP, how we started, who we are, and what we're doing and some of the challenges we're facing. Next slide, please. So the history of the group. So it was back in 2019, and even a little bit before, that we realized there was this unmet need for a GCEP to curate evidence for gene-disease relationships involved in syndromic disorders. So traditionally, as you all know, ClinGen has focused on creating GCEPs and VCEPs that look at a single organ system. Now, I'm from Illumina, part of the clinical laboratory, and we use gene curation as part of our workflow interpretation and reporting. So if we encounter a gene without curation data, we curate it as part of our turnaround time. We're very passionate about sharing our data and realized that there wasn't anywhere that at the time the GenCC was not in existence, there wasn't anywhere that we could actually share the gene curation data and want to get it out there. So approached ClinGen, who listened, and I'm delighted to say accepted this changing concept. So we are a very different GCEP in that sense, and we finally launched in March 2020. And in August 2022, we were awarded an NIH U24 grant that's going to fund us for the next few years, which is fantastic. We're very grateful. So we are funded, as I said, to look at the gene disease relationships that are with these syndromic presentations that involve multiple body systems. This is very different, and this has presented us with some challenges. And in fact, we have been challenged on occasion as to, can such a GCEP exist? And I'm hoping we can show you that it can. So the aims that we set up were to curate evidence for these GDRs for commonly tested syndromic disorders, to curate gene disease pairs identified through clinical exome or genetic sequencing performed by diagnostic labs from people within the SDGCEP, to curate recently reported GDRs from the Centre for Mendelian Genomics, and also to lend our expertise at the request of other GCEPs if they encounter genes that when they're linked in, they look more syndromic in presentations, so see what we could do to help. Next slide, please. Thank you. So I'm not going to run through all of the members. I have 2 slides of our beautiful photographs. Apologies, there are some still blank. So Anne and I co-chair. We have Ellie as our current coordinator. We've had some fantastic past coordinators who are up at the top left. And then this slide shows our 25 curators who come from a range of institutes and with a range of backgrounds. Sorry, yes. And then the next slide is our experts. We currently have 22 members, and we have our past members indicated at the bottom right. And again, these are from a whole range of experience.
Can you be near to the microphone a little bit more, because voice is very faint?
Okay, is that better?
Yeah.
Thank you for saying that. And we have some ClinGen leadership. We have OMIM represented. We have several diagnostic lab represented, people who are framework experts, people who are lab directors, and people who have expertise in a whole range of disorders. So if we could move to the next slide. So this map illustrates how geographically diverse we are, and the fact that we have 58 members at 28 different institutions across 8 different countries. So we really are trying to recruit and involve people from all over the world. This does raise challenges. We have to accommodate meetings over 17 time zones, I counted this morning, if I counted that appropriately. So if we go from GMT, you're going to sort of like -8 to +9. So we have meeting times that accommodate that. So on Wednesdays, we meet monthly, and you can see that that's sort of really prime time for Pacific time or Eastern time. Fridays, when we meet monthly, it's a little bit earlier, so that helps us in Europe. And then we have a quarterly meeting on Tuesdays, which really helps those out in Asia and Australasia. So hopefully everybody can make at least one of these meetings. And we do have good meeting attendance. Obviously, if you can see those different times of meetings, not everybody comes forward meetings, but that's perfectly acceptable, hence the different meeting times.
We actually get a lot of people from Australia joining the 6 a.m. call. I think less than the 10 p.m. call. And we wanted the SGT there is Singapore to make sure we had some times that accommodated that region of the world.
No, we do. People do show a lot of dedication. Now, I'm going to apologize in advance for this slide if I've missed anything. I didn't have a chance to proofread it, but I wanted to illustrate that a lot of our members are members of at least one other GCEP and or VCEP. So we have members in 23 different additional GCEPs and 17 VCEPs, and we cover 9 of the 16 clinical domain working groups. So this is showing the breadth of experience and the other GCEPs that we're directly interacting with through our membership. And members of other GCEPs are able to sort of bring back comments when we have genes or diseases that they've looked at in their GCEPs, and similarly they're able to give our advice. So I think this broad membership and broad sort of membership across the ClinGen GCEPs is one of our strengths. Next slide, please. So we don't fit into one of the traditional clinical domain working groups. So we're actually under other. And under other at the moment is the general gene curation expert panel, the prenatal gene curation expert panel, and then us. And I think that's a really good group. I think there's going to be a lot of overlap between the prenatal gene curation expert panel. We in fact have, I think, 3 members in common, and us. So yes, it's just we're currently lumped under other. So we'll have to see if we do something about coming up with a better name for that as we go on. Next slide, please. This is over to Anne. Thank you.
Yeah, so this is a lot covered in the intro slide, but we just recognized, again, many syndromic gene disease relationships were not covered by the current curation efforts. And so we really wanted to have a group that was really framework experts that had expertise in managing syndromic disorders that could really take on these multisystemic conditions. And so we thought this would really be a help in general to the community, get these genes curated, but also a help to the other GCEPs that might feel less comfortable when they start to go into genes that maybe have a single system or predominantly one system presentation, but then might have another presentation that was much more complicated, multisystemic. So we really have general expertise, and we tried to have broad expertise in the people we've recruited to the GCEP. And so we think we can meet the gaps that are occurring to get some more genes curated across syndromic disorders. So the first thing we did as a group one of the first things we did, in addition to just kicking off gene curations, was to make a scoring guidance to try to get us all in line and develop consistency in our practices and scoring. This was really inspired by the ID Autism GCEP, who was kind enough to lend us their scoring guidance. And so we took a look at that and put together some very similar sections in ours, and then had some particular areas around syndromic presentations that we wanted to address. And so I'm just listing all the different sections here. I'm going to go through a few of them. They are very text-heavy slides, but the majority of this is we won't cover today, but if anyone wants to see a copy of our guidance, we're happy to share it. So Courtney Thaxton very nicely worked on model systems for us. We preferred when the model organism is a mouse or rats, but most typically we're seeing mouse models. This is better recapitulation of phenotypes comparison to humans than some of the other models potentially, although we score other models in addition. And so we wanted to say when there was a mouse model, it really needed we need to be looking at the variant that matched with the variants we're seeing in the patient. So we wanted to be careful if we were seeing a missense variant in a patient, like all the variants reported in patients were missense variants, and there was heterozygous missense variants, and it was a homozygous knockout in a mouse. That's not really the same thing. And so that shouldn't be weighted the same as if you saw something that was the same variant, the same inheritance mode or same type of variant in a patient, and then that could be considered to have more points. It was also really important to have recapitulation of at least multiple of the phenotypes seen in the syndrome. The mouse may not have 100% of the phenotypes that have been described in humans, and it might also be that the mouse may not have been assessed for all the possible phenotypes, but we really wanted to see more than one phenotype if possible, and otherwise we would score partial points, but not the full maximum points. So again, typically if there was just a little bit of phenotype overlap, we would only consider it worth 1 point. And then sometimes we would have multiple mouse models and you can kind of add things over with that. For the zebrafish models, we prefer CRISPR modeling, but I did allow Morpholino strategy to be used because that's what a lot of the zebrafish model data is. There needed to be controls to ensure specificity of the knockout to the gene. We paid attention to whether there was a rescue with zebrafish or human RNA that was used. And if the rescue was zebrafish, the zebrafish allele not human, we downgrade by 0.5 points. And then I just want to highlight having the right experts and particularly having them actively participating in the GCEPs is a huge resource. And so we had invited Steve Murray from the Jackson Laboratory, who has worked on the Knockout Mouse Project and many mouse models, to join the GCEP. And he's on almost every call. And he's just an immense resource in his ability to drill down and really explain the mouse models to us. And if we don't see a phenotype, explain that maybe that one's not so measurable in mice, or we often don't see that in mice. And it's been just a great joy to have him, such an engaged member of our group. For functional alterations, this was designed by Amanda Klaus and Julie Taylor. We looked at patient cells. They must be derived from patients to fall into this category, and they must have been well characterized, and other genetic factors need to have been ruled out. And then we look at a functional alteration. We thought a lot about the cellular phenotype. And again, does that phenotype match at least one of the phenotypes we're seeing in the human, and not just necessarily wanting to show any alteration in a cell, but an alteration that makes sense for the syndromic presentations, or that involves a biochemical pathway that we know is associated with the disease phenotype. For non-patient cells to be scored the default, the assay must be performed using cell types that are in some way related to the disease pathway. And the evidence from non-specific cell types should be downgraded from default or not scored. And we often do list this data, but not provide scoring for it. These are still, I would say, giving us some wiggle room on this. And I think one of the things Alison and I have come away with is as we have discussion and debate, healthy debate on the calls, wanting to come back to this guidance and try to tighten it up even more to help us be consistent over time. So that's all I'm going to talk about for the guidance today. I'm just going to go into the aims in a little more detail of what we mean, how we put together the genes we've been curating from each of these lists. So the first is curate the evidence for gene disease relationships for the most commonly tested syndromic disorders. So that's very easy to say, but how do you figure out what those are? So we went to the genetic testing registry and we just asked how many panels a gene was on. It's kind of a rough and dirty way to just sort of guesstimate how often a gene is tested. We don't know how often each panel is ordered, but still just give us a sense of how often we're seeing these come up in testing. And then we looked at how many pathogenic or likely pathogenic variants in ClinVar for each gene, because we obviously it would be less interesting to curate a gene that is on 100 panels, but there's 2 ClinVar pathogenic variants, although sometimes that's because they're gain of function. So we wouldn't exclude those, but we just didn't prioritize them through this means. So we required there to be at least 5 ClinVar pathogenic or likely pathogenic variants. We just sorted by the number of panels, and then we went through and looked about which ones looked syndromic in their presentations and confirmed that the gene was not already registered in the gene tracker. And then as an extension of this, when we had conditions that came up, one of them, Myers-Gorlin syndrome genes, came up on this list as commonly tested. We said, well, actually, it probably makes sense to go through and complete this series. There's Myers-Gorlin syndromes 1 through 7. Let's actually go through all of those gene disease relationships and curate them all. So we've been adding things to our list that way also. The second is that we wanted to curate the gene-disease relationships that are coming from clinical exome or genome sequencing and diagnostic laboratories. And as an aim, this is very broad, but what we really mean is the diagnostic laboratories are doing a lot of this gene curation work in parallel as part of their individual genetic testing efforts. And a lot of this now has been shared in the GenCC, which is a fantastic resource. But at the time, this started before the GenCC, and also it's very, ClinGen is still obviously viewed as a higher level of scrutiny that has been applied to all of these curations. And so we wanted to be able to move as much of the work that's already happening in the diagnostic space into the ClinGen space, as opposed to like starting from scratch, just build on what's already being done. And many of the members of our Syndromic Disorders GCEP are curators at diagnostic testing laboratories. So we've been able to have them update their curations whenever it was last done for clinical testing, and then put it all into the GCI, present it on a call and do the full curation and get it approved by ClinGen. This has mainly been piloted by Illumina thus far, but we're hoping to increase these efforts as the other SDGCEP members and biocurators have bandwidth to do that. And we again have good representation from Ambry and Invitae also participating. We're also curating gene disease relationships from the Centers for Mendelian Genomics and other research efforts. This is sort of the same concept. So myself, I co-direct the Broad Institute Center for Mendelian Genomics with Heidi Ream. We obviously, as you can imagine, Heidi holds us to a high standard. So any genetic return, we make sure we actually, if there's already data in GenCC, we go with that. But if the gene has not been curated or does not meet moderate, strong, or definitive, we take another look at it. We do the full curation in-house. And so again, just trying to get these into ClinGen and not have that effort be lost, or not share. We want to share as much of the effort as we can. So we take the workflow from things that are coming through our research sequencing, and some of these are more recent gene discoveries, and we'll talk a little bit more about that later, but some of these are actually just genes that have already been reported that we're finding additional cases of. But it's really taking things that are coming through the research realm and pushing them through. And we also have representation on the GCEP from other researchers in the gene discovery space. We do think it's also important to highlight limited GDRs, particularly if the initial report was a while ago, because these are areas where there needs to be more evidence and publications. And so a lot of the times in syndromic disorders, we feel like we're not saying that this relationship isn't true. We're just saying we really just need more evidence. People need to publish more. There might be a fair number of variants in ClinVar, but we're not seeing the actual reports in the literature that we really need to establish this gene disease relationship. We think that's an important addition. So to date, we have, as a group, done 110 pre-curations or curations completed. You can see that about a third of our gene, actually maybe about half of our genes, not very good at math this morning, almost half of our genes are requiring a pre-curation. Things that are syndromic often are a little more complicated. They can involve multiple syndromes. They can involve a single system disorder and a multi-system disorder. And so we do spend a fair amount of effort on pre-curations. Our group really tries to, as following ClinGen guidance, lump things whenever possible. But there are many times where the phenotypes are distinct, or sometimes the molecular mechanisms are distinct enough that we feel like it really needs to be split into two entities. And we try to be thoughtful about this and make sure we're not splitting it in a way that is creating a lot of limited gene disease relationship curations. So while we try to lump whenever possible, we're actually slightly more prone to splitting as a group. And this is just the nature of the conditions that we're seeing. For the 76 curations that we've done to date, the vast majority have been definitive. There's a lot of well-established gene disease relationships out there that need to be added to these databases. Some are strong and hopefully with recuration and more time will become definitive. There are some that are moderate that I think are highlighting, again, where more effort is needed. And we did have another 8 that were limited. Some of these are new disease gene discoveries, but actually the vast majority are older, and it's just that additional data is needed. They're from like a case report or a very small case series initially with some plus or minus functional data. back to Alison.
Thank you. So this is a very colourful graph, is something we put together to illustrate exactly how multi-system and syndromic the disorders we're curating are. So these results from our sort of years 1 to 2 of our curation only. And this is where we've taken, so each bar represents a gene, and then the phenotypes associated with the disorder that we curated for have been coloured according to the HPO terms along the bottom. So this does provide a very colourful, stripy graph. As you can see, some are more syndromic than others, but most of them involve at least 3 different systems. So these really are crossing the traditional GCEPs, and hence why we think it's good for them to be curated with us. But it's also showing that we're having to take into account phenotypes across all of these different syndromes. Hence, I'm kind of repeating what we said, hence the need for experts who are used to looking at these coming through the clinic and signing out reports. But also, hopefully this illustrates one of our challenges in looking at these. But yeah, this was really just showed how syndromic the majority of these really are when you look at them. Next slide, please. And this is a slide that has, so that if you concentrate on the graph on the left, so this is showing the genetic evidence in blue and then the experimental evidence in green that we have scored for each of these genes. So the graph is split into 3. So at the bottom, that would be the limit on for limited. The sort of middle bar is where things would score moderate, and then above is strong or definitive. We've grouped them into AIM1, AIM2, and AIM3 genes. And I think it shows that they're not really a lot of difference between how we're able to score these genes. There are 4 that are starred, and these are the only 4 out of this group where we didn't use animal model data. And if you look on the right, there's a bar chart. So for 91% of the GDRs, there was an animal model available. And that's graph C shows the range of models. As we said, we by far use mouse as the most common, then zebrafish, but we do use others, C. elegans, Drosophila, Xenopus, and then others. Some, I think we've had a dog, I think we've had a pig, I think we've had a sheep. We've had sort of various animals, but most do have animal models available, and where we can, we score those. So we do score across the breadth of the genetic evidence available and the experimental evidence available, but would always try to find the animal model. That obviously gets more points, but it's also more convincing if it happens to match the phenotype. So the next slide, please. This is me. So we just wanted to talk a little bit about our interaction with other GCEPs. So it's very clear to us that the philosophy that ClinGen has of doing collaborative collaborations is really useful. We really appreciate ClinGen's collaborative nature. And as I said, we have membership across multiple other GCEPs, and we have both reached out to people and had people reach out to us. And I think the next slide, we have a figure showing this, where, so this graph illustrates where we have either shared or split curations. There are probably a couple of more since we made this graph, but our philosophy is that organ-specific GCEPs can curate, even if the gene's on our list, if an organ-specific GCEP feels it's most appropriate for them, then we will give it to them. And we will curate any genes that GCEPs think are more appropriate to be curated by us. We will share some curations, we'll provide secondary curations, and I have both gone and attended some of the GCEPs meetings just to give some of our help and explain how we've looked at the syndromic presentation. And I think this is a really healthy exchange between a wide range of GCEPs that certainly proved very useful for us. The next slide, please. This is Blue to you.
So I think we feel like things are going pretty well, but there have been some challenges that the group has encountered. We have a slide on each of these challenges, so I'm not going to read through those now. One is the question of when to curate new gene-disease relationships. I mentioned that some of the gene discovery work that we're doing isn't necessarily make sense to highlight after like the first very report of something, although we sometimes do if we have the information available. But we also just took a look at discoveries that we have discoveries made after 2019 and the time it took us to get to curating them on our list. So you can see some were very long after, still, several years after the discovery. Some were fairly short after the discovery. And the discovery is really the date that the publication came out with the first report. And so one of the things you'll notice is, this one, so I have them ordered in the time to curation here. So the longest is at the top. And you're seeing some limited things kind of near the top. There is one definitive here too. But the biggest thing is there's no like real strong pattern here. It's not like if you curate things right after they come out that there's necessarily a problem. And it's not like waiting a few years is necessarily going to solve that problem. So we really, I think it's a lot more the number of papers you're seeing, but one good matchmaker exchange paper with a lot of cases and some functional data can get you to strong. It just depends on what the data is in the paper. So we haven't really been able to use just the time since discovery to guide this. And so it's still something where there's no like systematic approach. We're just kind of trying to be thoughtful about including some of these, but not putting too much effort into these. But again, if you wanted to look at the number of papers and like a quick review or search, I think that would help you some, because there is a reasonable correlation with the number of papers we're seeing. Okay, one of the things we spent some time debating was gene disease relationships with a single recurrent variant. And our example here is PCGF2, which is caused where a variant at a specific position, that position 65, missense variants here cause Turnpenny-Fry syndrome, which has a distinct facial features and other multi-syndromic presentation. And so this became a big debate because we felt there was very clear evidence that, so I'm sure there's 3 variants here. These 3 variants are all at the position 65. They just cause different nucleotide changes within that codon. They do cause, I think, 2, maybe 3 different missense changes at that position 65. And basically, if you saw a missense variant or loss function variant, any other variant anywhere else in the gene, you couldn't really say much about it because no other variants in the gene have been found to be pathogenic. And these are almost all de novo. And there's like 20 patients with this variant. It's been seen many, many times. And so we were able to just still cure. We didn't want to like limit the gene disease relationship evidence because then it wouldn't necessarily make it onto the right testing panels. So we were able to say that the evidence, I believe, was strong in this case. Might be definitive. I can't remember the time frame. But we put in a sentence in the summary to say the impact of other than this Pro65 variants in PCGF2 is unknown, and given the single position involved to date, other variants would need very careful consideration. And this has been how we are addressing the situation, which I think is not unique to this condition, but other gain of function conditions may have only one or only a handful of variants. And so we're not necessarily, I guess this is true of everything though, for when you're talking about variants, you have to look at each variant individually. This one raised particular concern around the group, and I think we've largely come up with a solution. Okay, a topic I've heard that many groups are struggling with a little bit is the limited classification and listing it as disputed if the genetic evidence is less than 1, which you're most likely to get into if you're having missense variants reported, given loss of function variants often can get you over 1 pretty easily. So we had one curation that had 1.2 points, and this actually initially had less than 1 point, but we had more evidence in the experimental, and we ended up moving it because it was on the patient's specific variants into the genetic evidence. And so that's how we ended up going to 1.2 points on the genetic. The gene was NCAP-G2. It's a condensin. It is reported or reputed to cause Con-Katsanis syndrome. It's autosomal recessive neurodevelopmental syndrome with variant involvement of ocular, renal, skeletal, and sometimes cardiac systems, so definitely syndromic. There were 2 patients that have been reported in the literature with one had a compound heterozygous missense variant. The other had a homozygous missense variant that was a different missense variant than either of the 2 reported in the first patient. There was extensive functional data on the gene, but a lot of that is showing that the gene is very important for mitosis as opposed to like specific patient phenotypes. And then they had some evidence on the variant. So we overall as a group felt that this amounted to limited evidence. But again, initially we're concerned that we were needing to say it was disputed, and we really didn't feel that having less genetic evidence in this case, and in many cases, made us have concerns about it. We thought that it likely was going to work out, that this was going to be a gene for this condition, and that just more cases were needed, and that probably like more loss of function variants were likely lethal, which is why we weren't seeing more of the spectrum, although obviously very speculative. And so we were very comfortable calling this limited, but we just did not want to have to put a disputed tag on it. And again, we addressed that by getting the points, genetic points up to 1.2, which I think was the appropriate way these points should be applied. But we just wanted to highlight this for the group of thinking about when you do have genetic evidence loss of 1, that we might need a range of limited classifications, and you don't necessarily want to do anything to these types of gene disease relationships that would make anyone overlook this in genetic testing. Whereas if you put a disputed tag on a GDR, that might happen. And so just wanted to highlight caution there. Alison?
So naming challenges. So I think naming challenges is something we all face. There's the quote from Shakespeare, "What's in a name?" There's an awful lot in the name. I think we spend a lot of time debating names. We do a lot of lumping in our gene disease relationships, which requires us to come up with a new name. So when you are lumping, though, that can lead to challenges in conveying the full phenotype and not ending up with a really long, meaningless name. So often the name that we reach is a compromise, and we haven't had 100% consensus. So we're preferring to use dyadic names where possible. We're not renaming well-known syndromes. We're very appreciative of the new guidance that's come from ClinGen, but also aware that that's aimed at providing guidance for curation of GDRs rather than a condition name for a patient's diagnostic report. I think the two can be considered separate. We have heard discussion where it's not so much the actual name is important, but making sure you've got the right description of what you are curating. I do think the name is important because that's what people will go forward to use. So I'm sure that other GCEPs have challenges. I think it's a particular challenge for us where we're not dealing with a single organ. And I think we're going to sort of dig into the ClinGen naming guidance, but I think we may have to dig into that and come up with some additional suggestions of our own, or look back at our examples and see what's worked and hasn't worked. But just really wanted to have a slide to bring to you that this is a challenge that we face almost on a weekly basis in getting agreement. We're always very aligned on our curations on the scoring, and we have excellent presentations from our curators, and we tend to spend a good 5 to 10 minutes on deciding what to name it, which is important. But I think as a group and perhaps as a whole, we can do more work towards improving that decision making. So another challenge that we have is consistency in scoring. So I really do want to praise our document and all the work that everybody in the team put into producing it for our scoring document. It's been really helpful to ensure consistency in scoring across the GCEP, especially because we have our very rich membership who come from other GCEPs who may score things differently. We refer to it very often, and we do actively make sure that any discussions we've had on calls, particularly where any confusion has arisen about, oh, how are we scoring this? We identify places and go back and refine the documentation to make sure there aren't inconsistencies. And if we have identified something that perhaps our thinking has changed, then we've gone back to previous curations and ensured that the scoring is still stands. Thinking about consistency scoring, I do think we do make every effort to make the scoring accurate. But I think also, I just want to sort of say that I really believe in the strength of the framework, that the odd 0.1 or 0.2 here or there, because the ranges are so broad and the framework is so strong, it's probably not going to make a difference to the final overall classification. But I think it's this concept of getting consistency, particularly then if you're scoring gene disease relationships that don't have much evidence, you haven't got much to score. And I think, this is just something we address with our document, but again, it comes up time and time again in discussions with these curations. And sometimes when you curate genes from different disease groups and different disease types, they can present different challenges as different evidence. So We have produced the document, but it is a living organic document. We do refer to it, but also refine it. Next slide, please. That's you, Anne.
Yeah, so this is another challenge we've had. I don't know if other groups are coming across this. We have conditions that are predominantly recessive, so biallelic, but that there are some dominant variants that we think act in a dominant negative manner. And so there's really no inheritance mode that can currently supported in the GCI that addresses this situation. So again, it's often recessive with a small number of monoallelic pathogenic variants. It's come up on 2 genes that it's actually currently a blocker for us, so Trex and PLCB4. One proposed solution had been there's semi-dominant as an option in the GCI. However, this is really not a semi-dominant situation. In semi-dominant, the carriers of the recessive variants should have a phenotype. It might be a more mild form of the phenotype, but they should have some phenotype. But this is really the pathogenic dominant variants are typically de novo and not inherited from an unaffected parent or otherwise inherited from an affected parent, and really acting in a dominant negative way. So kind of like recessive in that they're impacting both alleles or both copies of the protein at the end of the day. So one solution would be to split them. However, there's often just, there's not a good reason to do that. And the phenotypes typically aren't distinct, although sometimes there's a little bit of severity difference between them, but it's typically often a spectrum. Most of the time, these variants are either dominant or recessive. There's not like variants that could cause both forms. So they are really like a spectrum going from recessive to dominant negative in the phenotype and everything. And if the biggest downside of splitting them out is it's because it's often just a few cases that have been reported as dominant, they often will end up limited. And so we think that could be confusing also. And mostly it's not consistent with our other lumping practices to split things like that out. So one proposal we've had that would need buy-in from somebody larger in ClinGen is to add an inheritance mode option in the GCI for an autosomal recessive / dominant that really let us take these situations into account. And then we could explain a little bit more what's going on in these individual cases in the evidence summaries. So that's something we'd love to see. This probably isn't the forum to ask for it until we'll sort that out of where we should make that request. Back to Alison.
So what are we doing now? What are our future directions? So we're going to continue curating our A1, 2, and 3 genes. We're planning to start doing some recurations. So we're going to look and identify the ones that would appear to be most suitable, or the, so probably more towards the limited and moderate end, to see if there's any more evidence been published that we can bump those up. We are going to we're going to expand on the number of expedited curations. We've got a goal of doing 12 of those a year, and I've got a slide explaining what we mean by expedited curations. Again, this is something relatively new, but we really do have a long gene list, and we hope this is something that could help us to get through more genes in a year. Currently, we review a pre-curations or curations per meeting. So having an additional 12 that we can review outside of meetings could really help bump our numbers up. We're going to begin writing a manuscript on our experience and what we found. And just wanted to point out that we have been asked a couple of times. We have no plans for syndromic VSEP at the moment, given the breadth of conditions. It'd be nearly impossible to come up with sort of VSEP level guidance. So that is not a future direction. We wanted to mention it because it's the question that comes up. And I think most GCEPs, when they're going for a couple of years, do start a VSEP. So if we just have a look at what we mean by expedited curation.
Also on the VSEP plan, I would add that if other groups are interested in starting VSEPs around like certain specific syndromic disorders or groups of disorders, we had talked about like chromatin disorders, one could sort of organize a VSEP in this space. So it's a very open, unmet need right now. So encourage anyone who's interested in meeting that need to go for it. And we're happy to kind of help out in an advisory way, but it's not something we have any plans to do.
Yeah, sorry, I should have made that very clear, but yes, we're not going to do it personally. So expedited curations are full curations. The difference is the review process is somewhat expedited. So we are reviewing the curation, approving it offline, by 2 of the experts from the panel. So these are appropriate for genes that don't need a formal pre-curation and relatively straightforward. So we have 3 examples here where they are going to definitely be definitive. The evidence should be fairly straightforward and there'll be plenty of it. And the idea is that we would share curations by email, do a quick formal review in a meeting. So we could review up to 6 of these in one meeting. So the group gets a chance to see the curations and raise any issues and answer any questions and do a formal vote on the classification. Anne and I have done 2 of these so far, and they were relatively easy to do, a good use of time. So we're going to sort of expand on the number of these that we do. So asking our curators to do them and send to us for review offline. The next slide, please. So that last slide, just say thank you for your attention. A special thank you to our curators and experts for their work over the years, to Alicia, Ellie and Vanessa for helping with bringing things together for the slides for today's presentation. And I wanted to add a special thank you to Marina and Jenny, without whom we wouldn't have got off the ground in the first place. So big thank you to them. And yes, does anybody have any questions?
There was the one question in the chat, but I think, Anne, you possibly covered that when you were talking about the overlap with the CMG work. But Anna, did you want to comment as to whether you thought that was adequately answered? Anna, not Anne.
It was good, thanks. I didn't know if you would cover it, and so I just wanted to throw it in there, and it was well covered later. Thank you.
Excellent. I guess one of the other things we sort of like that I almost thought that was getting at was on some of these limited ones where you do see ClinVar variants, or like where we know there are other cases in researchers' hands that we're collaborating with or that we've sequenced ourselves, the ability to kind of count those in the curations. And thus far, we have stayed away from that with the hopes that just pointing out where people need to publish additional things. But that is something that in the future might be helpful to get some of these more limited, moderate curations up to a higher level.
Are there any other questions from anyone else on the call? If not, then I guess we can all have 15 minutes back to our day. So thank you so much to Anne and Alison for presenting that on behalf of the Syndromic group. It's nice to see how far you guys have come, having started off with this idea of there is no one covering syndromic disorders to really filling that niche area. And also I particularly appreciated the challenges you presented, because they're faced by lots of the groups within ClinGen. So I wouldn't be surprised if you have some people reaching out afterwards for a bit more guidance on how syndromic is approaching them. But with that, we will finish up. Our next meeting is June 14th, I want to say. I don't have it in front of me. The second Wednesday in June, and it'll be a presentation from one of our hereditary cancer GCEP. So following this theme of just experiencing what some of the other groups within ClinGen is doing. I hope everyone has a wonderful day.
Thanks for having us.
Thank you.
Thank you.